Hello, everybody. Today I have an amazing chat with Nick Lane, who is a biochemist who thinks in incredibly deep ways about the origin of life, the emergence of mitochondria, and everything there. And I think that the beautiful, beautiful piece that Nick has is he comes at these things instead of from the frame of information and DNA and RNA. He comes at things from the frame of energy and specifically the frame of like electrical gradients. And so I think it's if you want to understand how to think deeply about how life evolves and all of that, and especially if you want to look at it from this amazing kind of heterodox frame of electricity gradients, then Nick is a great person to listen to. So we chat about all that. We kind of go deep on the Krebs cycle in the middle, but I think it'll help you really understand these reframings of like how to actually think about how life evolves, and then at the end we get into some more big picture stuff. So I hope you enjoy it, and thank you and goodbye. Hello, Rhys's Pieces. I'm Rhys, the co-founder of Root, and welcome to The Rhys Show. This century is a turning point in human history, and I'm here to help you navigate it. I hope you come away with a new understanding of our scientific, technological, and societal trends that are poised to radically reshape our world, and how you can work with those trends to become a live player in building a solar punk future. And today, I'm excited to chat with Nick Lane. Nick is a British biochemist and writer. He's the professor of evolutionary biology at University College London and has authored many excellent books, including the most recent of which is Transformer: The Deep Chemistry of Life and Death. Nick, thanks for being on the show and welcome.
Thank you. Pleasure.
Yeah, excited to dive in. And I think there's Nick and I were kind of chatting about this before the show, where it's like there's a set of folks who are thinking in a really kind of deep, fundamental way about core processes that underlie all of life and how the universe evolves and all of that. Carl Friston and Michael Levin, etc. And so Nick is another amazing person in that set of amazing people. And so I kind of like want to dive into his brain and understand his view on kind of the origin of, and so Nick, just for you and for our listeners to know, the goal here is to kind of to tell a story about life and how it, what was happening before it started, how it's evolved, and then where we are today. How do humans play a role, and then like what might happen in the future. And I want to give, I know,
yes, not unambitious at all for an hour. Yes.
Exactly. And then as another note just for you is that the frame that I'll kind of be coming from is this frame that also the listeners will be coming from is I'm writing this book on what information wants, which is a big history of how the patterns of the universe emerge and how they evolve and how replicators create the tree of life and memes create the tree of ideas and what that means for us as a species. So with that, let's kind of dive into, I want to start at the beginning, but actually before the beginning, which is, we could start at the origin of life, and you've written, an amazing book on that. But tell me a little bit about, for you as a biochemist, how you think about the world before life. Like, do you think much about like pre-life happenings?
Well, yes, but not in a very coherent kind of way. I mean, I suppose you think about what was the planet like, and almost certainly there was no oxygen. So if you were to be there, you would asphyxiate immediately. You wouldn't enjoy it much. That's the first thing you'd realize is that you were going to die, and then it would be an ocean world. There would be a few islands, maybe a total land mass on the scale of Australia or something like that across the entire world, maybe smaller than that. We don't really know, but there's not a lot of land. And the moon is very close, and the tidal range is huge, and the waves are massive, and so on. It's a pretty disturbing kind of place to be. And most of the environments that I'm thinking about for the origin of life, mostly in this context, are down at the bottom of the ocean. Now, lots of people don't agree with this. Pretty much everything I'm going to say to you today, lots of people would not agree with it. So you're going to get a biased view, but it's my view, and you're talking to me, so that's too bad. But so I'm down at the bottom of the ocean, and we're dealing with hydrothermal systems. Now, if I say hydrothermal, most people would probably think of a kind of underwater volcano or something with lots of black smoke coming, belching out of these things. They're very visual. But there's also other types of vent which are kind of more gentle. They're not obviously venting anything. They're not smoking really very much. In fact, they are active. Sometimes they're called white smokers because there is some smoke and it's kind of white rather than black, but it's often there's nothing obvious coming out. And these are a reaction between water, just seawater, any water, it can be water on land as well, and rock. The cold rock, this rock that you get on the seafloor and down below that. And I sometimes say to the students in a lecture or something, you take a lump of this rock and you put it in a bucket, and it should bubble. You should get hydrogen bubbles coming off it, but you might have to wait a while for the first bubble. It's not a very exciting experiment to show anyone, but go down to the bottom of the ocean to the high pressure and higher temperatures and so on, and actually it's going to fizz. You're going to have it's going to effectively give you bubbling caustic soda, and that's the kind of thing that's giving rise to these vents. It's minerals like olivine that make up most of the mantle of the Earth. So this is the kind of the context. I'm imagining a dead planet, a kind of sterile, inorganic planet covered with an angry ocean, with lots of vents across the sea floor, with kind of bubbling caustic soda coming out of them, and so the question is, well, how does that give rise to life? And it's
And let's pause
yeah.
on that for a second because, and I want to, yeah, exactly, and I love your description of it because, and I love people call it the Hadean eon, where it's like it's Hades, it sucked,
Yeah, though that's
it's
changed a bit because we used to think it was amazingly hot as well, and people you say Hadean and people would think about lava flows, but probably there weren't so many lava flows at that point.
interesting. So it was colder. It was just normal temperature. Was it room
Yeah,
temp?
I mean that's probably the one thing you'd say is you could go for a swim in that sea, and I think it's unlikely that you. Some people think it's up at 70 degrees. I don't think that's very likely personally, but
Mhm.
70 degrees you'd burn. You wouldn't want to go for a swim in that, but it wouldn't be like going into a hot spring or something.
Yeah,
So
gosh.
and it may well be that it was down at 5 to 20 degrees or 30 degrees or something. These kind of numbers are really difficult to constrain.
Yeah,
I'm not a
yeah,
geologist, but geologists spend a lot of time trying to work out from fractionation of oxygen isotopes. There's ways of getting at these questions, but they're very difficult to interpret.
yeah.
And I think the
And
oceans were coolish.
yeah, okay, coolish. Yeah, that's a good. Yeah, it's like, so in Hadean eon, maybe we should call it the yeah, just like a wave pool instead. Like a big wave pool.
Yeah,
Where that's
yeah,
like kind of the
yeah.
feeling that I
That's
get.
probably close to
Um,
my imagination. Yeah.
Yeah, exactly. So, but actually, I want to push you on something here, but just before we talk, because you've done so much exciting research around, the abiogenesis, the start of life, and then mitochondria and all that. And I want to push you actually on the time between 14 billion and 4 billion years ago. Like that kind of how do you, as a biochemist, do you like when you imagine that time of the universe where the big thing happened and then the galaxies are forming and planets are like, how do you take does your biochemist lens like how does it think about that period?
It doesn't much, to be honest. I mean, it's fascinating. I like reading about physics. I like reading about cosmology, but it doesn't have any direct meaning to me. It would have direct meaning to some models of the origin of life. If you're thinking about cosmic chemistry, if you're thinking that what you really wanted to have here is organic material delivered on a meteorite coming from outer space somewhere, and cosmic chemistry, helium radicals, all this kind of thing that will generate you organic molecules, and they land on Earth, they land in the oceans, and you end up with a primordial soup or something. There's problems. I mean, well, it's true. This cosmic chemistry happens. It's real. It's not imaginary. And organics arrive on Earth on meteorites. It's definitely true. It definitely happens. It's conceivable and possible that bacteria or something arrive on Earth in that way as well, panspermia. Um, but it doesn't solve the problem of the origin of life because he just said, well, it happened somewhere else. We don't know where, we don't know when, we don't know how. We have now zero information. So instead, we take a kind of an assumption, and science is underpinned with assumptions. The assumption is it started here. You know, maybe it didn't, but we know it is here. Let's assume it started here. Is there anything we can say about the conditions? Is there anything about life on Earth that can guide us to how it might have started on Earth? And the
Great,
answer
great.
is, yeah, there's quite a lot. Um, and actually so strong that I would turn it around the other way and say, if we're looking for life elsewhere, there is a good grounding for looking for water, for looking for carbon chemistry and so on. It could have been other ways, but it works so well here, and there's so many good reasons to think it happened this way on Earth for a reason, for good chemical reasons and physical reasons, that we can extrapolate outwards and say, well, here's a kind of the basics of what we should be looking for.
Yeah, great. Yeah, that makes sense. Is that you can't have even the words like cosmic chemistry, where organics can be out there in the world, panspermia or whatever, where it's like yeah, where the things coming in like those are pop, but in general, the weight of evidence seems to be the other way. And I also like what you say there for a reason, because usually when people say, "Oh, something happened for a reason," it's like some kind of godlike reason or whatever. But then you're
Uh,
like, wait a second,
yeah,
it's
no,
a
I mean,
biological
I'm
or
thinking
chemical
of
reason. So you just consider that.
biological and chemical reasons.
Exactly.
Um,
It's just a funny I was
yeah,
just
yeah.
laughing at that. So tell me about okay, so we're at the start of Earth. We got this not the Hadean Earth, but maybe the big wave pool Earth. And you so how the hell did life start? What was going on down there?
Um, well, again, there's as many views as there are people working on the subject. And actually, an interesting point is that this is a clash of disciplines. It's a question in science, and so which sciences are relevant to the question? Well, the answer is physics is, and chemistry is, and biology is, and geology is, and cosmology is, and and these are disciplines that go back with their kind of intellectual way of approaching the question in the region of 100 200 years or so. And one thing you kind of realize working on the origin of life is that even though most people genuinely do their best to kind of cast off their discipline and think like a scientist, you can't help. Your training kind of gets in your way, and I'm a biologist and I think like a biologist, and I want to think like a physicist, but I'm in the end I'm constrained by the way that I think about the world, and I just have to deal with that. Um, it's actually great fun trying to be a physicist and a chemist and a biologist and a geologist all at the same time, because you're perpetually an excitable student going through areas of science that you kind of would love to know more about, and they pay me to know more about it. What's not to like? So um, I am coming from a biochemistry point of view, a biology point of view, and what I'm interested in is well, really, the origin of life then is the origin of cells, and it's the origin of bacterial cells, because those are the earliest forms of life. We can build a tree of life. We can try and reconstruct what were the earliest cells, what did they look like, how did they behave, what did they grow from, um, and that kind of gives us an end point. And the start point could be almost literally anywhere you want. So, but make another assumption. That other assumption is Occam's razor. Let's just take the simplest possible path. So, if the first cells that we know about grow from hydrogen and CO2, and they're autotrophic in that sense that they're growing from gases, they're not eating things, and they're powering that reaction. In fact, it turns out that they're powering it by electrical charge on membranes. That's in effect what a cell membrane is really doing. It's partly separating the inside from the outside, but it's got a charge, and that charge is driving all the work. And so then the question is, well, are there environments that are conducive to this kind of chemistry? Can we have somewhere which has got a cell-like structure with electrical charges on the barriers around it, with carbon dioxide and hydrogen? And the answer is, well, yes. Actually, the entire seafloor was covered in vents that did exactly that. Um, which is why I'm interested in the seafloor, why I'm interested in this. So it links up with biology just beautifully. Doesn't mean it happens. It's just two things, you can put them together and they look very beautiful side by side. And so you think, okay, well, how do I go from here to there? And it's a long distance. You could think, if you think about a bacterial cell, I don't know what people have in mind if you think about a bacterial cell, but I mean, it's a tiny speck of a thing that you can't even see. And if you look down an electron microscope, it's got some stuff inside it and you can't work out what it is. It's really almost nothing. But at a molecular level, it's got all these machines, molecular machines, and whirring parts, and long strands of DNA and informational code, and it's an astonishingly complex entity. And if you think about the kind of the evolutionary distance between a bacterial cell and a dinosaur or a human being, it's probably not as great as the distance between the bacterial cell and prebiotic chemistry, the simplest prebiotic chemistry. You're going back, you're crossing a huge distance. And to have any, and we have almost no information on how do you cross that distance. Some different people with their different backgrounds will think, okay, well, I'm going to prioritize information, or I'm going to prioritize metabolism, or membranes, or whatever it may be. And whatever you do, you've really got to try and think, okay, well, here's the first step, here's the second step, here's the next step. How am I going to join them up? I know the end point. The end point is a bacterial cell with ribosomes and DNA and molecular machines and things. And the starting point might be cyanide chemistry, or it might be CO2 and hydrogen. But how do I line up the steps and think of a test for each step? And that's
Beautiful.
effectively what we're doing in the lab. Is we've tried to join up the dots and then test the steps.
Yeah, and I'd love to hear about those dots in a second. But one thing I just love the kind of the perspective that you provided here, which is just like a pluralist, like interdisciplinary thing, where you're like you can put on the different hats, and sometimes you can put on the physics hat and try to see the things from the physics perspective, and you also know that kind of like your full expertise is in the biologist realm and the biochemistry realm, and so just knowing those different frames. And I think another brilliant thing you said is just like thinking about what to prioritize as well, where you're like, okay, you can either prioritize the information flows, how's information being, or you can prioritize the membranes, and like how the membranes are working, or you can like kind of like Friston style Markov blankets or whatever, or you can prioritize just the energy and the metabolism and something like the Krebs cycle. And so I think that is so you can kind of and then from each of those you can kind of construct the path.
Well, you can, but I think the problem has been in the field generally is that people bring their day job, which is to say you come in, your background is in molecular biology and RNA, and so what you're really interested in is an RNA world and the origin of information in an RNA world. And you just make an assumption that, well, what do you need for RNA? You need the nucleotide building blocks. Where do they come from? Well, they come from over there. We'll leave the chemists to sort out where they come from. And the chemists, well, they say, okay, so the molecular biologists, they need RNA, so we've got to make nucleotides. We'll go and make nucleotides, and they do that. So, okay, here's your nucleotides. But the kind of the joining up, that you're not only making nucleotides, you're also making lots of other stuff. It's hard to make nucleotides. It's much easier to make amino acids or fatty acids for membranes. If you can make nucleotides, you can make all of those other things as well in almost any environment. So how do they relate to each other? And if you've got an RNA world and information is invented in an RNA world, well, it's actually a really deep, serious problem. How does kind of random sequences of RNA invent information that goes into a whole metabolism? It's a big, big, serious question. And I think it's the wrong question because it's the wrong order.
Great. Let's double click on that again. I think that's a beautiful part about kind of the lens, because I mean, in some ways, I'm kind of priced into my book of what information wants. So I'm coming from the informational lens, and so I'm like blah 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 RNA world. You had a thing, and then somehow that thing. These autocatalytic cycles where survival of the fittest chemical reaction, and then like it emerged, and then those out. So but I would love to hear because you're saying energies first, Krebs cycle first, metabolism first. So tell us more about that kind of lens.
Yeah. So I suppose the first thing to say is if you're going to have a large number of nucleotides, you've got to have a system making those nucleotides. Wherever they come from, you've got to make the things. And if you're growing and you're copying and you're expanding, you're doubling all the time, then you can probably reuse some, but basically you're going to have to make lots of new ones. And where are they all coming from? And saying, well, they're coming from over there is too vague, really. It's not good enough. If people do an experiment on it and they just have a kind of 1 molar solution of nucleotides and don't worry about where they all came from. So
and let me pause you for a quick second, just for the listeners who, just in case aren't aware, that a nucleotide. So RNA. So there's DNA, and DNA has 2 strands. RNA is just 1 strand, and the nucleotide is like half of the RNA strand. So there's C, G, A, and T on these things, and technically RNA has a different set of 4, but it's roughly the same 4.
Sorry, yes, I should have said that. Yes.
No, you're fine. You're fine. You're fine. And so the nucleotides, like, how do you get the code? These like the
Yeah,
like
that's right.
half of the code,
So I
you
mean,
know. And
RNA is basically a long polymer of all of these letters. They're often called, but the nucleotides are the chemical building blocks, and you just string them all together. And with us, we have 3 billion of them in our own genetic code,
And correct
all
me if
strung
correct
up.
me if I'm wrong, but is a nucleotide, when I imagine nucleotide, was I correct in saying that it was you have an RNA thing, which an RNA molecule, or whatever I call it, this big string, and a nucleotide a single letter of that thing, a single
Yes, basically,
or
yes.
it is okay, and then a nucleoside is like that, but it's like without the what is a nucleoside then?
Oh, well, it's without the phosphate. So I mean, they're
Without
just
the
they're
phosphate.
just
Great, great, great.
technical terms, but
Great,
yes.
great, great, great. Okay, so nucleotides. So we're trying to build up this series of letters, and so you need to make a ton of them. So go back to like how do we? How does the energy? How do we get the energy to make all of them? Yeah.
Yeah. So if you use life as a guide to its own origin, and you say, well, the simplest cells are growing from CO2 and hydrogen, and it goes um, into basically a metabolism where if you react hydrogen and CO2 together, what you get is Krebs cycle intermediates. Now, Krebs cycle intermediates are uh, carboxylic acids, which is to say they're basically just carbon, hydrogen, and oxygen, and nothing else. And what all life does, it makes amino acids from those carboxylic acids. So you've got just the carbon, hydrogen, oxygen. You add in nitrogen as well. Um, and the nucleotide, the building blocks of RNA and DNA, well, they're made from amino acids and sugars, and the sugars are made from the Krebs cycle intermediates as well. They're also made from these carboxylic acids. So there's a kind of a conserved, universally conserved structure to all of metabolism. And it's interesting that the chemistry is universally conserved, but the genes are not. The genes that apparently encode this metabolism are different in bacteria and archaea. Not always different, sometimes they're the same, but there's these two fundamental domains of prokaryotic life, the bacteria and the archaea. They have quite different genes, they diverge very early on, and they have different genes catalyzing the same chemistry very often, which kind of says the chemistry came before the genes. It doesn't necessarily say that, but that's one interpretation of it. Now, that 5, 6, 7 years ago would have been a really hand wavy statement. Um, Effectively, what I'm saying is that this core of metabolism, starting with CO2 and hydrogen, going into Krebs cycle intermediates, into sugars, into amino acids, into nucleotides, will just happen spontaneously in the absence of any genes or enzymes or any, well, I was going to say information. There's entropic information in there. There's chemistry, there's thermodynamics, there's kinetics. There's a lot of information in there, but it's not a kind of genetic information. Um, so I say 5, 6 years ago it would have been hand wavy, but a lot of this has now been done in the lab. It really does work. You start with CO2 and hydrogen, you really do get Krebs cycle intermediates. You start with those, you really do get amino acids. You can get nucleotides, you can get sugars. You can now, it's still patchy. Some things have been done, others haven't. Some have been done under some conditions, others under different conditions. By no means do we really have a clear answer yet. But it's gone from being no way to wow, this is a real possibility that it really works this way. So let's just assume then that it does work that way, that we're capable of making these things. It's much harder because there's more steps involved to make the building blocks of RNA or DNA than it is to make fatty acids or amino acids. And fatty acids, they make up the membranes. And we and others have shown that if you just mix fatty acids in water and kind of shake it up a bit, you get what I like to call protocells, which is to say you get a membrane around an internal enclosed aqueous space. And they're capable, they're very dynamic. They can grow, they can divide, they can fuse together. They're constantly moving and doing things. They look quite lifelike, even though it's just a chemical system that you're looking at. And the amino acids, they do all kinds of amazing things as well. They interact with minerals and can convert them into things like iron sulfur clusters that are involved in fixing CO2 to make more organics, including more amino acids. So there's positive feedbacks. You mentioned autocatalytic loops. The positive feedback is an autocatalytic loop. Um, And so if you take this seriously and you start thinking through, okay, what steps do we expect to happen first? What would come afterwards? Nucleotides are quite late in the game, and to get to them, you've really got to. I mean, it was quite possible to think about replicating protocells with a proto-metabolism which is basically thermodynamically governed, that this stuff just happens spontaneously and is capable of making the nucleotides and is capable of getting better at making the nucleotides through positive feedbacks. So we end up with a kind of non-living system which are replicating protocells that are quite lifelike in many ways, but has no genetic information. It has some information in the membrane and in the metabolism, which is a thermodynamically encoded information, you might say, which says this is, and that's deterministic. That says you can do this, but you can't do that. And you're never going to be able to do that because thermodynamics says you can do this. If you've got a different catalyst, maybe you can do that, but where's that catalyst coming from? And what you end up with, if you then think, okay, now I've got these growing protocells. I'm going to introduce. I've got nucleotides forming in them. What happens if I allow them to polymerize for whatever reason? Now I have a random string of RNA, random letters, no information content whatsoever. But we know in the code that there are patterns in the code that suggest there's non-random interactions with amino acids. Hydrophobicity, for example, size is another one. That if you've got a random sequence of RNA, you will have interactions with amino acids that bind to that RNA for physical chemistry reasons. And if they bind, they're more likely to polymerize. And so you're templating from a random RNA sequence, you're templating a non-random peptide. And that non-random peptide will have function in a growing protocell. It will either help its growth or hinder its growth. And so from this point of view, I've just introduced information into biology without doing anything other than introducing a random sequence of RNA. And so the question of the origin of information goes back to the thermodynamic questions of why the hell did I just, where did all this chemistry, this prebiotic chemistry, proto-metabolic chemistry, why is it like that? Can we see the answer in carbon chemistry? Is it somewhere else? It's hard to say, but it's that's where
beautiful.
the information comes into biology, is in thermodynamics, I would say, which is why I have
Yeah,
to be a
that's interesting.
physicist.
Yeah, exactly. That's funny. That's funny. Yes, yeah. Then you have to get into thermodynamic information instead of
Yeah.
genetic information. Yeah. So what I'm hearing, which I think is interesting, is like, yeah, there's the RNA world hypothesis, blah blah blah. You got these little RNA things that already have these big old strings of nucleotides, and they're just kind of finding their own autocatalytic loops. And but the question is like, wait a second, how did all those nucleotides get just like randomly created? And so
And how do
then
they
so
know
what
what
you're
to
saying
do
is, hey,
next?
and how do they know what to do next? And so it's like, and so I think that there's what you can do is you can say, let's actually look at something more kind of, the energy side, something maybe more fundamental or kind of upstream of the nucleotides, which is when you combine, you got carbon, you got CO2 and hydrogen, and you get these, what was the term for the CHO stuff, the
Yes.
carbohydro,
So carboxylic acids.
carboxylic
Yeah.
acids, carboxylic
So a
acids,
lot of
those
people will
are
have heard of
carbon,
the Krebs
hydrogen, and
cycle. Yeah, they're really simple molecules. They may have between 2, 3, 4, 5, 6. I mean, they can have more than that, but the familiar ones are between 2 and 6 carbons with oxygens and hydrogens bound to them. So it's a string of carbons joined together, a string of 6, say, joined together with various oxygens and hydrogens added on.
yeah, great. So yeah, you're getting these little, and you can imagine for listening. And for me, it was really cool to learn about just like, yeah, how like both glucose and all these things are just and I forget the name of these things. Yeah, there's so much of life is just like these strings of 6 carbons that have hydrogens or oxygens like put in different places. And so what these carboxylic acids, it's like they can be 6 long. Maybe at the beginning they were lower. And in the Krebs cycle, and I guess the crucial bit is that what you're getting is you have CO2 and hydrogen that gives you these carboxylic acids, aka these little strings of carbon. You kind of like, you're mushing shit together, and of course it's gonna make some that's the stuff mushed together. And so then what you get is then those things then lead to amino acids and sugars, which then,
Yeah.
and that's the building blocks of a nucleotide.
I mean, I don't know how many people have heard of the Krebs cycle or are familiar with the Krebs cycle, but insofar as anyone's heard of it, what they're probably likely to be familiar with is the idea that it's something to do with generating energy in our own mitochondria. And that's
Exactly.
absolutely bang on. Um, so what's actually going on there? Well, what's happening in our own mitochondria is we're taking food. Glucose is the usual easy example to give, but it can be fats, it can be amino acids. There's lots of it, and they get fed into the Krebs cycle. They get broken down into carboxylic acids. So the Krebs cycle is made up of these carboxylic acids, and you start out fusing a very simple two fairly simple ones together to make a 6-carbon string. It's called citric acid, and sometimes it's called the citric acid cycle, or sometimes the tricarboxylic acid cycle. But anyway, you got this molecule. It's 6 carbons long. It's got lots of oxygens and hydrogens bound to it. And then effectively what the Krebs cycle does is, well, it yanks out a carbon dioxide. It yanks out a carbon, one of the carbons with 2 oxygens on it. That we breathe it out is waste. And then it yanks out a hydrogen or 2 hydrogens. And now those hydrogens are attached to other molecules, NAD, for example, nicotinamide. I always get my names muddled up, but adenine dinucleotide. But the point is, it's hydrogen. We're dealing with hydrogen atoms. And you can think of it as hydrogen gas almost, but it's never hydrogen gas. We're dealing with hydrogen atoms stuck to other things. And in respiration, we feed these hydrogen atoms to oxygen. That's basically what we're doing. We're burning hydrogen in oxygen. It's rocket fuel. We're powered by rocket fuel. So what happens? We're not releasing all that energy as heat. We're actually conserving it. And in effect, we're splitting the hydrogen atom into an electron and a proton. So a hydrogen atom is basically it. It's one electron, one proton. The electron gets fed to oxygen. The proton goes across the membrane. The energy that's released powers the extrusion of a proton across the membrane. This sounds very technical, but basically what you've done is you've just put a positive charge over there and left a negative charge over here. And so you've just generated an electrical membrane potential. You've got a voltage across that membrane. So we've got an electrified membrane now, and that's what you're doing. So the Krebs cycle is spinning around, it's stripping out CO2, it's stripping out hydrogen, it's burning the hydrogen and oxygen and using the power to electrify the membrane. It's kind of as simple as that. Now, that's what we do. That's what all animals do. It's what a lot of bacteria do, but some bacteria do the opposite. And the older we go, so we're feeding the hydrogen to oxygen, but what happened before there was any oxygen? Well, think about, we've got a cycle that's going around, and it's stripping out CO2 and hydrogen to electrify a membrane. Now, in a hydrothermal vent where you've got lots of hydrogen and CO2, well, and you have electrified barriers around protocells, then we actually have the opposite. We now have a reverse Krebs cycle. It's taking CO2 and hydrogen, it's binding them together to make these things. It's doing it repeatedly, and it's being powered by the electrical charge on the membrane. So it's a reverse Krebs cycle, and it turns gases into the fabric of life. It's fantastic.
Whoa. Okay. So let me understand. So yeah, that's great. Thank you for that understanding. Yeah, and so like exactly, my understanding of Krebs cycle is just like it is the thing that produces or I guess it's
ATP,
it's the thing that produces
yes.
ATP, because what it does is it takes you could get the membrane, you make a voltage across it, and then when you try to when you do the ATP creation process, it kind of that is where the energy comes from, in order to like take the
Yeah.
ADP and like push it a little,
So the
processing
ATP
onto
is usually called the universal energy currency.
yeah,
And
yeah,
I
yeah,
like to think of it as like a coin that you put in. So a protein is like a slot machine, I suppose, and the ATP is like a coin, and you put the coin in the slot machine, and it does what it does. It changes its state momentarily.
yeah, yeah,
In a protein,
yeah,
it changes its conformation when ATP binds to it or something, and then
yeah,
it hydrolyzes, it splits off, so you have ADP and a phosphate that goes over there, and the protein changes its shape again. So
yeah.
basically life is powered that way, but then the question becomes, well, where does all the ATP come from? And the answer is, well, ATP synthesis is driven by this electrical charge on the membrane. And the
Yeah,
electrical
which is
charge on the
created by the
membrane
Krebs
is driven
cycle.
by
Yeah,
respiration, which is from burning
yeah.
food in oxygen. So you don't have any
Which is
ATP.
created by the Krebs. I guess, or the Krebs cycle is the thing that needs as input. It needs I guess, yeah, the Krebs cycle takes as input these glucose and puts as output ATP. Is that
Well,
right?
yes, it is. That's basically right. But that way of seeing it kind of clouds the issue, because
Okay.
really what it's doing, the Krebs cycle as such, is pulling out hydrogen and CO2. And the hydrogen is being
Yeah.
fed to oxygen to electrify the membrane. That membrane now is used to make ATP, but it's
Yeah.
used for lots of other things as well. And in bacteria, that electrically charged membrane is used to fix CO2. It's used to take CO2 and convert it into organic molecules. It's driven by the electrical charge on the membrane. And so
Hmm.
the bits that I kind of want you to focus on is not the ATP so much as the carbon dioxide, the hydrogen, and the charge on the membrane. And those are the working parts that really matter. And you can spin it one way, and you're effectively burning hydrogen in oxygen to generate a charge, and you spin it the other way, and you're taking in hydrogen, binding it onto CO2, and using the electrical charge to power that. So it's exactly the opposite.
Yeah.
And
And so,
that's what's
let's say,
beautiful about these vents is they've got an electrical charge, they've got CO2, they've got hydrogen, and you've got the system which is converting gases, inorganic gases from a sterile planet into the building blocks of life.
cool. Okay, I think I understand. Which is that, yeah, at the beginning we have these non-building blocks of life. We don't have amino acids. We don't have sugars. And what we need to get is we need to get those things, and those things are the a thing that can create those is a reverse Krebs cycle.
That's
Is
exactly
that
it.
right?
That's the
Yeah,
one.
exactly. And so the it's great. I feel like I'm I'm in like your office hours right now. Um, and so
I I hope everyone else doesn't feel that way as well.
no, 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 it's good, it's good. And so, yeah, the reverse Krebs cycle, and I guess the crucial, and that's an interesting. I do think that that reframing is really helpful, which is like ATP is like we kind of see ATP as a core building block, obviously a core energy, kind of currency of life. It's like actually to go deeper and to think about the cycle itself, to think about the hydrogen, the carbon, and the oxygen, and to think about. And I guess what I'm hearing is that at the beginning of life, what we had was the reverse of the Krebs cycle. We had these energy gradients that are, or sorry, these electron gradients that
Mhm.
already existed,
Well,
voltages, and
proton
what
gradients, really.
proton
Yeah.
gradients exactly, and what you get is you can go the instead of using that to turn a thing to create ATP synthase to create ATP, what you can instead do is go the reverse, which is you already kind of have the energy,
Well, the
and
energy
then
is there in the charge, in the proton gradients. So
yeah,
in our own mitochondria, we are using an electrically charged membrane to power ATP
yeah,
synthesis. But
yeah,
forget about ATP. It's kind of almost
yeah,
not relevant here. What we're
no,
interested in
yeah,
is the charge that can power work. And the
yes.
work that we're interested in it powering in the reverse Krebs cycle is the reaction of hydrogen and CO2.
Yes. Yes. Great. 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 Exactly. And so, and then that reaction then builds those building blocks of. So, I mean, this is a great. I think in the key crucial thing for me, I do want to move to like the kind of the next part of the cycle,
Yeah.
which is the like mitochondria and etc. But I think it's a great. I mean, I think that this understanding and that there's a whole big thing here around just like how we think about the origin of life and also energy and how we think about these energy gradients and that. So do think it's a very crucial frame on this. And also, I think you also there's information in kind of the simple kind of genetic information sense, and then there's information in like the thermodynamic sense. And so, kind of thinking in terms of information in the thermodynamic sense at the beginning it probably is better. Okay, so let's transition into. So now we have these. Now we actually have something that does look like the RNA world, but there was something before it. Obviously, the reverse Krebs cycle that kind of created it. And now we have these autocatalytic survival fittest reaction, whatever it might be. I guess what I would love to hear is like when you think about I guess, yeah. What's my question here? Yeah. So we go from that to, and we think about the kind of evolution of biology over time, from the deep sea vents, obviously, then to getting photosynthesis and respiration and mitochondria, and then to multicellular organisms and humans. How do you kind of think about? I mean, this is a tough question. How do you think about that big journey of that 4 billion years to us, and how like energy plays a role in it?
Yeah. I mean, there's a few things to say there. I mean, one of them is at the origin of life. I was talking about the end point is bacteria, and bacteria are amazingly tiny and seem to be very simple, but in terms of the molecular machinery, they're extraordinarily complex and amazingly highly evolved. Now, how long does that take? I don't know. I don't think it necessarily takes all that long, because we're talking about chemistry mostly, and chemistry has to happen quickly or it's not going to happen at all. I don't think we're dealing with things that build up and accumulate and crystallize in an environment and sit there and wait. If we're talking about autotrophic chemistry, we're talking about reactions that are happening in nanoseconds or milliseconds or seconds, maybe minutes at the slowest, but otherwise it's just gone, especially if you're in a hydrothermal vent where everything's flushing through all the time. So I think it can happen pretty quickly. But the reason that most people would say, "Oh, it's going to take millions of years," is because of the kind of the sheer distance that we've got to cross, the innovation distance of going from prebiotic chemistry to complex cells. And we tend to think of evolution as slow because what happens next is, well, we have 2 billion years of nothing really. It's just bacteria. We get through at the end of that 2 billion year period of just bacteria, we get to what's called the Great Oxidation Event. And around that time, there's a snowball Earth as well. So this is a kind of middle age crisis of the Earth, where in fact the snowball Earth means that the sea froze over at the equator and possibly even sealed off the entire planet, so it was just completely sealed in ice. Some people talk about a slush ball Earth where it was not necessarily completely sealed, but it was a catastrophic change in climate and conditions, and that was linked with oxygen and photosynthesis. So we know that photosynthesis arose around that time. Maybe it could have been as much as 1 billion years before and just somehow didn't make a mark for very long, but it could have happened almost immediately before as well. And then after that, it kind of gets back into a stasis that's called the boring billion very often. So we have another billion years. And now in this time, the complex eukaryotic cell arose. We don't really know exactly when. So eukaryotes, that's our own kind of cell. They're a lot bigger, 10 to 100,000 times larger than bacteria in volume. And with a large nucleus where all the DNA is stored, and then all these moving parts. If you look at a eukaryotic cell down a microscope, it's full of membranes, it's full of sacs and compartments, and you don't need to know what any of this stuff is to realize that there's a lot of stuff there which you tend not to see in bacteria. And if you look at one of your own cells down a microscope, or a plant cell, or a mushroom cell, or an amoeba, they've all got the same stuff. They've all got the same, not only they have a nucleus, eukaryotic means true nucleus. So they all have this nucleus, but the nucleus is surrounded by a membrane. It's the same structure in the membrane. They've got pores with the same proteins that make up the pores. They've got an elastic lamina, which is exactly the same. They've got what's called an endoplasmic reticulum, a kind of a network of membranes inside cells. They've got mitochondria. All our cells are basically the same. You would hardly tell the difference between a plant cell and an animal cell down a microscope, except for kind of the vacuole or something trivial like that. So here's a huge gap. I like to think of it as the black hole at the heart of evolution, because bacteria arose, and archaea, very similar to bacteria in their appearance and their morphology, but not in their genetics. They arose 4 billion years ago, and the reason that we look at fossil relics of what look a lot like bacteria 4 billion years ago, we think they are bacteria because bacteria look like that now. They haven't changed. So actually, if you take the eukaryotes out of the picture, bacteria just did 4 billion years as a flat line. They didn't change at all, and archaea as well. Now, that's interesting from an information point of view because they have large populations, they have strong selection, they have searched genetic sequence space exhaustively, far more than eukaryotes ever did, and yet they stayed as small, fairly simple cells. They got stuck in a rut, and eukaryotes broke out of that rut, and it happened abruptly. And this is one of the things. It goes back. I mean, it goes back a long way. But Stephen Jay Gould talked about punctuated equilibrium. Evolution can happen really quickly. It's just that most of the time it's stuck in a rut. The conditions are kind of basically not conducive to change. Now, the fact that bacteria and archaea explored all this sequence space and did not come up with complexity, morphological complexity, to me says there was a constraint. There was a constraint that was not a genetic constraint. And I think it was a constraint linked with energy. It's partly genetics, and here's how. What we know about eukaryotes is they all have mitochondria, the power packs and cells that are generating this electrically charged membrane where the Krebs cycle is happening. And all eukaryotes have these, and apart from a few that lost it, but they lost it later on. And all mitochondria have their own genes. They've got a little outpost of genes that are necessary for effectively controlling this electrical charge. So, I can't let this charge go. It's right at the heart of my thinking about life. We've got electrical fields on membranes, and they're driving an awful lot of stuff. And you've got a gene outpost right next to this electrical charges field. And if you get rid of it, you'll die. This is the mitochondrial DNA. And these were bacteria once. They were free living bacteria that got trapped inside other cells. It seems to be a pretty unusual thing to happen. And once you're in there, in effect, it's hard to leave again. You kind of and it's a very intimate and very conflicting relationship. I mean, this is you think of Sartre's idea of hell as being trapped in a house with one person or something for eternity. I mean, I'm not sure I would see that as hell necessarily, but in the case of the mitochondria, you've just trapped a population of bacteria with their own agenda and their own interests, if you like, inside a host cell which has got a different agenda and a different set of interests. And you say, right, you are forever together. Figure it out. And they did, and the way they figured it out was to come up with a nucleus and an endomembrane system and all the rest of this stuff. It was all really about how do you deal with your pesky endosymbionts. So what happens? Well, you end up with multibacterial power. So each of these mitochondria have got the same electrically charged membranes that they ever did. They're contributing this power to the cause. They've kept a few genes, but most of the genes they got rid of. They're left with, in our own case, 38 genes in the mitochondria. And I think the record is Reclinomonas americanus, which has got about 98 genes in its mitochondria. But basically, from 3,000 or 4,000 in a bacterium, you're down to less than 100. And the overhead costs of copying them and making proteins from them is nothing. But you've got all this energy. And that means you can swell up your nuclear genome to become enormous. And that's basically what we see in modern eukaryotic cells. We have a genetic asymmetry where we have a huge nuclear genome encoding 20,000 genes, but a lot of regulatory capacity as well, lots of regulatory RNA and whatever it may be, supported energetically by these tiny mitochondrial genomes. So it's a genomic asymmetry that we have. And that in itself then allows multicellular life of a sophistication like us, because in effect, there is nothing out there with the complexity of a flea composed of bacterial cells. There's simple multicellularity in bacteria, which is to say there's differentiation from the same genome. You have different phenotypes from the same genotype, but it's pretty simple. If you want to have something as complex as a human being with multiple different tissues and organs and so on, without genetic conflict, so if you were to try and build a human from bacterial cells and do it as a kind of as a biofilm, as a bacterial mat, where different bacteria effectively lived in your head and different ones lived in your heart and different ones lived in your kidneys, they'd all basically be at war with each other. They're genetically different to each other. They have a different agenda. It will fall to pieces very quickly. What we actually have is clonal cells. We are a giant clone of one cell type that came from a fertilized egg. So the DNA in all our cells is the same, but in the brain we switch on these genes and we switch off those genes, and in the heart we switch on a different set of genes, and in the lungs a different set. And that means we need to have all of these sets of genes sitting in the nucleus and resistant to decay, resistant to mutations. We've got to have a large genome that doesn't decompose. And to do that, you have to have a lot of energy, and you need to have sex, which is basically the mechanism for quality control of large genomes.
Yeah. Yeah. This, I mean, and let me, so this is, I love this perspective because it really, when I think about, um, the kind of transition into life and again coming from my like information first lens, I think about the start of the the mitochondria coming out and hanging out with the trying to be with its friend and being enveloped by the and they're hanging out, and I mostly think of it from the perspective of, oh, now the in the past the gene had to be attached to the cell, the DNA had to be attached to the cell wall, but now it can live freely in the nucleus, and that increases the length of the DNA by like roughly 1 million times. And that is amazing because then you can like, it's just like getting a new kind of USB drive where you're like, oh my God, now I can actually like code up a bunch of more amazing things. And so from an information first lens, I'm like, oh, that's what matters, is the bigger drive. And what I'm hearing is that is kind of true, but there's also a what really matters is that you have this amazing power cell that only has 38 genes in it that is just able to power it's able to power the rest of, obviously it powers that, like these multicellular lives that can be that are these clonal things where both the information lens, we're all kind of in alignment, where it's like it's all one gene and there's one kind of
Yeah.
output. Yeah, you go.
No, I mean, this idea that the bacterial genome is circular and is attached to the cell membrane and so on, it's a nice idea. It's quite an old idea.
Yeah,
And it kind of it's similar in the sense that it's a structural constraint. So what I'm talking about, why do bacteria remain bacterial? I'm saying there's a structural constraint. They need an endosymbiont. They need a power pack. They need
yeah,
mitochondria. But there are other structural constraints that people have talked about. One of them is the cell wall. You have a cell wall, so you can't really just grow big very easily, or whatever it may be. If you lose the cell wall, you're probably going to die. But if you somehow survive, then maybe you'll be all right. That's it was a theory that goes back to Tom Cavalier-Smith about phagocytosis, that you can't eat, engulf another cell unless you lose the cell wall. But there are plenty of archaea and bacteria that don't have a cell wall, and they don't become miraculously large and phagocytic and anything. And there are also bacteria that have multiple chromosomes and straight chromosomes and chromosomes that are not attached to the cell wall. And again, they don't show any sign of becoming large and complex. So it's not that structural constraint. That's not what's holding bacteria back. I completely agree with you that with a small bacterial genome, whatever the constraints are, you just can't do all this stuff. You need a large genome, and you need to be able to maintain that genome. And the maintenance of that genome, there's two things you need really. One of them is the energy to just store it and express it, because it costs a lot of energy. If you've got 20,000 genes and you want to make 100,000 proteins from each gene, there's an energy bill that you've got to pay. And if you can't pay that energy bill, you're going to die and selection will be rid of you. So that's one thing. But the other thing is, if you're just a bacterial cell and you scale up to a eukaryotic sized genome, and you continue to do your replication bacterial style, either by cloning yourself or by what's called lateral gene transfer, where you pick up bits of DNA from the environment. If you're stressed, you pick up some DNA and stick it into your genome and hope it'll do the trick. And it doesn't do the trick, pick up another bit and another bit, and you keep on going. It works pretty well for small genomes, but the bigger the genome gets, the harder it is to maintain the quality of the genome by picking up bits of DNA from the environment. You've basically got to spend your entire life picking up random bits of DNA and hoping that one of them will work. And it doesn't. I mean, we can show by mathematical modeling that it doesn't work. What sex does in eukaryotes, it uses the same machinery that we see in homologous recombination in bacteria. It's exactly the same genes and enzymes involved. We know where it came from. But instead of picking up bits of DNA from the environment, we fuse two cells together. In our own case, the sperm and the egg, but it goes back to single-celled eukaryotes that basically produce sperm-like things, motile gametes that swim around and fuse together. And then when they fuse together, they line up all the chromosomes. And then they double everything, and then they do lots of crossing over, recombination between the chromosomes. And then they go through 2 rounds of cell division, which is called a 2-step meiosis, and they produce gametes again that way. So what have we just seen? We've just seen that instead of it being piecemeal bits of DNA from the environment, we have fused with another cell. So we've kind of compromised our individuality as a cell and become a fused entity. And we've systematically and reciprocally lined up all the chromosomes and recombined across all of them. So it's both systematic and it's reciprocal. And that's what we don't see in bacteria. And again, to have a large genome, you must do something like that. You can't do it any other way. And it's a pretty major change to force on a cell, is to just kind of fuse with another cell and share its DNA like that. I mean, it's a big
It's a
shift.
big ask.
Yeah.
Yeah. That's great. And so as we get into wrap mode here, I mean, so this has been a great, I mean, to learn about the just like kind of just a reframe around energy within these systems, and that really, and I loved how you said also later where it's like like the primary thing you think about is these electrical gradients and like these voltage gradients, and so you're like, okay, great, and that using that lens to kind of explore life and biological life is like a really good, and both at the start and when we got eukaryotes and to today is like a really good frame. And so I think, you know, trying to apply that also to like current humans and future, I think will be an interesting exercise. It also is really helpful for me just to hear about the like the different kind of constraints that exist within these systems, where you can either think of them as you're doing this, you should think about informational constraints while you're also thinking about these kind of like energetic constraints. And so that's kind of a very helpful lens for me. As a wrap for the listeners, check out, I mean, yeah, Nick has a bunch of amazing books. I mean, they're quite excellent. The most recent one is called Transformer: The Deep Chemistry of Life and Death. He also has this amazing one called The Vital Question, which on energy, evolution, and the origins of complex life. And then Life Ascending: The 10 Great Inventions of Evolution. It's good. You know, it's great stuff. He's also, what were you going to say?
Oh, no, thank you. It's just kind of you to
Yeah,
plug
yeah.
that.
Yeah, I mean, I love shilling stuff. And correct me if I'm wrong, but you're not on Twitter. Is that right?
Yeah, that's right.
Oh
I'm
my
too
God.
obsessive to kind of. I wouldn't be able to let it go, and I would
Yeah.
cease. I would cease functioning in other ways that I care more about. So I'm deliberately not on Twitter.
That makes sense. Well, you can definitely go check him out at nick-lane.net or exploring these books. Is there anything else you want to say to our listeners to kind of wrap up today, Nick?
Well, one thing we've talked about early origin of life and early evolution and the eukaryotic cell, and we touched on multicellularity and the origin of animals and so on. But the last half of the the last third or so of the book is really about aging and the way that the Krebs cycle and these electrical charges on membranes run down, and what goes wrong as we get older, and why it's linked to diseases like cancer and so on. And then there's an epilogue, which I barely dare to talk about in the time remaining, but it's really about how do electrical fields play into consciousness, and not consciousness necessarily. There's an awful lot going on in consciousness, but I tend to try to simplify things down to the lowest possible bar that I can see, and the lowest possible bar about what a feeling is, about what a qualia is, to me, is linked with electrical fields, and that's not original to me at all. But why? Well, if you think that the first cells had an electrical charge and electrical fields on their membranes, and that it was linked to effectively the metabolic health of that cell, and it integrates. Metabolism is billions, literally billions of reactions per second, second after second after second after second, going on all the time. So how does one reaction know what another reaction's doing? This is not at the level of genetic regulation. There's too much stuff happening to regulate that whole system with genes alone. It's linked through the Krebs cycle to the electrical charge on the membrane, and all of metabolism, because it's linked to the Krebs cycle, we now have it's linked in series to the charge on the membrane. And if that charge fluctuates, then all of metabolism shifts. And that has a feedback effect on the activity of genes and so on. So there's a lovely way of seeing the simplest form, if you like, the stream of consciousness, which is linked to electrical fields on the self that is a bacterium.
I love that. I love that. That's a great. And what it makes me think of is just like, and kind of like what we were doing before, where it's like, you can either think of information as genetic information, you know, or you can think of it in kind of a thermodynamic sense and kind of entropy and whatever, that kind of physics information. And then this is a similar thing for me, where it's like you can think about, oh, how are all of these things doing gene regulating? And the simple answer is, you know, something like gene regulation and blah blah blah, it's in the DNA. But then there's and the lens that you're providing, a lens that like I'm thinking of, so like Michael Levin adjacent things. It's like, how is electricity moving through the system, and how are these electrical fields moving through the system, and that lens actually provides lots of juicy answers.
I think so. I mean, I'm not knocking genes. Obviously, genes are central to
Some of
biology
your
and
best friends
important.
are genes. Some of your best
Well,
friends are genes.
but it's not the only thing going on in biology,
Yeah.
and it's quite, there's a long distance. The idea of electrical fields being important in biology, it goes back a long way. And you
Yeah.
mentioned Mike Levin a couple of times. I mean, his work is transforming the way that people are seeing this kind of thing, and he's thinking about it in terms of neural networks and in terms of development and the electrical fields linking cells together, and bioelectric fields and patterning and so on. And he's thinking mostly about the cell membranes. I'm thinking mainly coming from inside the cells, inside the mitochondria, where the fields are even stronger. And how do they interact with fields on the membranes? And how does all of this interact with the genes? They're really big, open questions. And it's not to say genes are not important. Obviously they are, but they're not the only things. They're not the only guys in town. There's other stuff
Yeah.
going on which is important too.
I love it. I love it. Well, thank you again for your time today, Nick. And definitely, listeners, if you want to go check out this kind of electric field, first view on life, then check out any of Nick's books, and they're delightful and juicy. And thank you for listening, everybody. Goodbye.
Thank you.
Thanks so much for listening today. If you liked the show, please give us a 5-star podcast review or subscribe on YouTube. And if you'd like to chat about this episode with a community of amazing, smart, ambitious, divergent people, come on by and join our Discord. You can find it at root.co. That's ROOTE.co. And then finally, if you'd like to contribute to these ideas being shared more widely in society, you can support the podcast production team at patreon.com/Rhys Lindmark. That's patreon.com/RHYSLINDMARK. Thanks, and see you here for the next episode. Bye.